Hi there. In this video, I'll talk about multicellularity and cell communication. Multicellularity was a major innovation in evolution. But once you have more than one cell and cell type, the cells have to somehow communicate to know how to grow, to know what to do. It required a little more sophistication in how to deal with the outside environment, including what cell was next to what other cell. Here's a complete tree of life showing the major groups of organisms, including bacteria, archaea, the protists, plants, amoeba, and opisthecons. And the dark shading indicates the number of multicellular organisms or species in each group, and the white areas indicate the unicellular species and organisms in each of those groups. So for bacteria, archaea, and most of the protist eukaryotes, most of the species are unicellular, and only a few groups have some multicellular members. But then by the time you get to the plants, amoebae, and fungi and animals, most of the organisms are now multicellular. So if we just focus on the animal clade, where most of the organisms are multicellular, down here are the only unicellular organisms, which are called choanoflagellates, and those are animal cells. And then working up through the anemone and sponges, and then off onto this line, which includes the chordates, humans, and some other more primitive organisms, up here into some of the worms, nematodes, and then insects and crustaceans up here. So there's a really wide range of body types and lifestyles in this group. So as I indicated, the cells have to somehow communicate within a multicellular organism. So there's what's called paracrine signaling, where there are molecules secreted by certain cells which signal other cells to do certain functions. There are also cells connected with one another that communicate directly. And then there are other cells, synaptic signaling in neurons, where one cell sends out a chemical neurotransmitter, which is picked up by the other cell, and that's transmitted down that cell as well to propagate a nerve impulse. There's also endocrine signaling, which is long range. So this would be epinephrine being secreted that then that chemical flows through the bloodstream and interacts with target cells. It includes insulin, which is released by the pancreas and then goes into the bloodstream and signals cells to take up sugars. And there are other types of signaling as well. So in essence, each cell has to know where it is, what it's doing, what it's next to. And so that takes quite a few genes, and that's taken a lot of evolutionary time to develop those systems. These signaling mechanisms actually developed first in bacteria. Even though they're unicellular, they would signal other individual cells of the same species. It's called quorum sensing in bacteria. So in bacterial cells, they would produce a certain inducer molecule, which would be secreted by those cells. And then if the concentration is low, it wouldn't turn on these targeting genes. But as concentrations rose, especially when the cells aggregated with other cells of the same species, then that inducer molecule would attach to a receptor or response element inside the cell, and it would turn on target genes to express certain proteins and mechanisms and functions. Another type of signaling is when the bacterium produces a certain chemical that then is secreted out of the cell and interacts with a surface protein. And the surface protein, once you get sufficient concentrations of that, it will release another molecule that induces a promoter to turn on certain genes and gene products. Here are some specific examples. Here's a bacterium that produces what's called a lactone, a sugar-type molecule that then is excreted from the cell. Other cells will pick it up, or the same cell will pick it up. If it's in high enough concentration, it attaches to a response element that becomes a promoter to turn on specific gene products. Here's another one which produces a signal peptide that goes through an ABC transporter that then is picked up by a serine kinase receptor on the outside. The serine kinase then phosphorylates a certain protein which interacts with a promoter molecule that increases transcription of specific genes, and then there's a certain response from the molecules that it forms. And finally, this one down here, which is more typical of some of the response elements. So this one forms several different types of molecules that are released from the cell and then interact with several different receptor proteins on the cell surface. And then those send signals through to different proteins inside the cell that then interact to either turn on or turn off specific genes and their gene products. And one of these is luciferase, and this forms a protein that makes flashes of light. You can see this sometimes out in the ocean, and sailors for hundreds of years have noticed this, that when you pass over certain parts of the ocean or you see waves sometimes, they fluoresce green and sometimes blue colors. At night you can see these. And these are caused by this luciferase, which is caused by aggregation or high concentrations of these cells, which are producing high concentrations of these reactive molecules that then turn on the genes to make luciferase, which produces the flashes of light from those bacteria. So even in bacteria, unicellular organisms, they are able to communicate with other bacterial cells of the same species. And some bacteria actually are multicellular or have at least two different cell types. One of them is Caulobacter crescentus. And in this case, the Caulobacter has two different forms, a swarmer cell, which is motile, and then a stalked cell, which is non-motile. So the stalked cell sticks itself down to a rock surface or the surface on the bottom of a stream, and then starts to divide. But the two cells are asymmetric. So one cell actually grows a flagellum, and once they separate, it swims away. Whereas the stalked cell stays in one place and doesn't move again, although it keeps forming more swarmer cells. The swarmer cells, on the other hand, find a suitable surface and begin growing a stalk and lose their flagella, and then they become stalk cells. So this all requires a pretty large number of genes and decisions by those cells on what the conditions are and which genes to turn on and which genes to turn off. And all of this, of course, is done by molecular means. In plants and animals, multicellularity is much more complex, and the communications are therefore much more complex as well. So in green algal evolution, and green algae lead to higher plants, by the way, so you have first Chlamydomonas, where you have single cells, individual cells, although they are able to communicate, and they do have reproduction where a plus type gets together with a minus type to form a zygote. And then you have other species that start to aggregate and to communicate more. And then you have balls of cells that are embedded within a jelly type substance, and those are communicating. And then larger aggregations of cells together, and again, they're all communicating mainly to photosynthesize and to share resources, etc. And then by the time you get to this type of cell, and then Volvox, which is the larger cell aggregation multicellular organism, then you have specialization of cells on the outside versus on the inside. And each one of these steps then has to be built upon by the previous steps so that you then have a communication system that works for each one of those cell types and species. Animals also, most, as I said, are multicellular. And the cells also communicate in some complex developmental patterns. And these are all vertebrates here shown. And there's a vertebrate pattern and patterning that is set up very early during development, so that you know right away as development occurs, and these are all just a few millimeters in length, what type of organism is going to be formed, and it's clear that it's a vertebrate. At a later stage, then other more specific genes and gene products turn on, eventually leading to a form that's more characteristic for each one of those animals, and then further development into an adult of that type of organism due to other communications from cell to cell and long range from cell to cell. Cells communicate with the outside world by receptors on the outer surface of those cells, and this pertains to animals and plants. And then there are certain either molecules or other receptors that attach cell to cell. Then that receptor sends a particular signal into the cell that turns on or off other functions within the cell, and that what's called a signal transduction pathway, that is a transfer of that information from one protein to another, leads to certain things happening in those cells, including changes in gene expression, apoptosis, that is cell death, sensor of certain things within the cell, sensors of DNA damage, sensors for whether they're being invaded by pathogens or by something else coming from the outside of the cell. And I won't go into it in much more detail in this class, but all of these ones that are in this orange font, each one of those, if they're mutated, has been implicated in certain types of cancer. So a lot of the signal transduction pathways, cell death pathways, changes in DNA expression, monitoring DNA damage. If those are mutated, often they'll lead to tumors and cancers. So that's one thing that goes wrong in multicellularity. If the cells do not communicate well, if they lose their contact with the outside, they'll often go out of control and become a cancer. That is, they're not able to sense what's around them and react accordingly. So these are some of those categories of proteins and genes that, if they're mutated, can cause cancer or at least loss of control of that cell. And so these have evolved over a period of hundreds of millions of years in these multicellular organisms, including some of these growth factors out here, the receptors for those growth factors, and contacts with other cells, knowing that you're contacting another cell. And then these signal transducers, these proteins that from one to the next together signal something else to happen in the cell, including apoptosis, that is cell death, changes in gene expression, changes in the cell cycle, or realizing which part of the cell cycle they're in, things that recognize DNA damage, etc. So these have all evolved with multicellularity in order to form an organized organism. That is, if you have disorganization, then you don't have a multicellular organism that is functional. One part of being a multicellular organism is that there is a developmental process. And so this shows a bit of development of an amphibian, a frog, where you have, even in the egg cell, a segregation between different molecules from one end of the cell to the other end of the cell. And then you start getting cell divisions, where the cell will actually start producing more cells of certain types, and one type will interact with another type. So when you first have the initial stages of development, you get a blastocele formed, where part of the cells will become the ectoderm, primarily epidermis and things like that. And then future endoderm, internal organs, and then portions of the ectoderm and endoderm interact to form new types of cells, which are known as mesoderm, and those form specialized cells within the internal parts of the organism. Later, there's an invagination to form the initial parts of the gut, and you can see how the ectoderm, mesoderm, and endoderm are interacting with one another to make sure that they're forming the right type of cell for that particular part of development, which will eventually lead to development of more and more cells, tissues, and a complete functioning organism. Later in development, some of the mesoderm cells become what are called mesenchyme cells. These cells are able to move around in certain portions of the organism to influence other cells by signaling them to perform certain functions, including gastrulation. That is, the secondary mesenchyme here will start to influence the cells here of the ectoderm and endoderm, and also cause those to invaginate or grow inward to form the initial portions of the gut. So those are some cells that are influencing other types of cells. These types of cells have been studied in all sorts of organisms, including simple animals going up to very complex animals. And you can see that the complexity of these cells and their interactions with other cells have changed through evolutionary time and become more sophisticated and more exacting in forming those more complex organisms. For example, in sponges, the developmental pathway is quite simple. There are very few different types of cells, but then by the time you get to mammals and insects, then the number of those genes and the way that they interact is much greater, and the way that they affect the cells is much more refined, and the diversity of cells is greater as well. So by the time you get to a fully developed organism, going from zygote to blastocele to gastrula, then you can find where each one of those tissue types is going to wind up in the adult organism. For instance, the ectoderm becomes the skin cells of the epidermis, gut cells, and neurons. Then the mesoderm become muscle cells, tube cells of the kidney, red blood cells, smooth muscle in the gut, etc. And the endoderm becomes pancreatic cells, some types of pancreatic cells, some thyroid cells, and certain types of lung cells. And then there are also germ cells that are set aside very early in development so they don't go through many replication cycles, which would cause more mutations. And if you just focus on blood cells, then you start with these multipotential hemopoietic stem cells. And remember, all of these cells start out with the same genome, but then it's expressed in different ways. Different genes are turned on at different times, depending on how it's being signaled from the outside. So you can get a myeloid cell, which then these differentiate and mature into erythrocytes, mast cells, as well as basophils, neutrophils, eosinophils, monocytes, and macrophages, and then megakaryocytes that form thrombocytes for clotting. And then there are lymphoid progenitor cells, which when they mature become natural killer T cells. Small lymphocytes, which become either B lymphocytes or T lymphocytes, and the B lymphocytes, some of them become plasma cells. So remember, all of these start out with the same genome, but then by the time they get down to here, sometimes their genomes have actually been changed and altered, especially in lymphocytes. But they differentiate into different cell types based on how they're being signaled to go down those pathways. And so this indicates that these multicellular organisms are highly complex on how the genes are controlled and when they're turned on. And this has been a part of an evolutionary pathway that is literally hundreds of millions of years old. Also, when some of these cells are altered in certain ways, they can become cancer cells and take over the body by dividing uncontrollably, so that at first it's sort of minor. They're dividing uncontrollably, but it doesn't hurt the organism so much until it starts invading other tissues and destroying them, and then it becomes life-threatening. So now I want to take a look at a multicellular organism and how it functions and how it develops, showing sort of an evolutionary but a developmental pattern also. And the first one I want to talk about is Cenorhabditis elegans, which is a nematode. And there are two sexes. There is a male, and then there's a hermaphrodite, which has both male and female sex organs. So it actually can fertilize its own eggs and doesn't need a male. And there are really about 1,000 hermaphrodites to every male, so there are a lot more hermaphrodites. So it develops from a zygote, and then those cells divide and divide further, finally forming something that's a rudimentary nematode, which then grows into the adult here. So here are the two sexes, the hermaphrodite up above, which consists of 959 somatic cells. People have actually mapped these out, plus sperm and egg. So here's a pretty simple structure. There's the mouth, the pharynx, the intestines, and then the anus down here. And then the rest of the animal is pretty much made up of sex organs to produce oocytes and sperm, and then to eject the fertilized eggs, or to lay the fertilized eggs. The male is here. It's much simpler. Has only male sex organs to produce sperm, and it's made up of 1,031 somatic cells plus many sperm cells. And as I said, the hermaphrodite can go on just producing eggs and pretty much almost clones of herself, and doesn't mate with the male very often. In fact, as I said, there's about 1,000 hermaphrodites to every male, so many more hermaphrodites are present in a population. I should mention also that there are some species of Cenorhabditis that appear to not have had any males present for at least 15 million years. So they've been producing as hermaphrodites only for at least 15 million years. So as I said, they've studied these nematodes in great detail, and they have actually looked at the cell fates. And if you look in the embryonic stage here, all the cell divisions that are known, going from the zygote to this many minutes in the development of that organism. And then you go into the post-embryonic stage and produce a few more types of cells. Some cells are actually interacting with other cells during those cell divisions. And then there are certain ones here marked in red, blue, and green, where in every cell division, one of the cells goes on to become something and the other cell dies. And it's a programmed process called apoptosis, or programmed cell death. And this is very common in multicellular organisms, where one cell is induced to go down a certain developmental pathway, and the other cell dies, and it's part of that cell division process that leads to programmed cell death. So during evolution, this is a very common thing that happens in multicellular organisms, including humans. So this is one example of that in neuron development. In the males, they have certain neurons called male-specific chemotaxis cells that allow them to detect the hermaphrodite and where she is, or he is, and then lead it to the hermaphrodite so that it can mate with the hermaphrodite. However, the hermaphrodite doesn't need to find another hermaphrodite to mate with, so that those organisms don't need those neurons, and they don't develop because there's programmed cell death at an early point. So those neurons never develop in the hermaphrodite. Alternatively, the hermaphrodites need a specific neuron, or a set of neurons, called hermaphrodite-specific egg-laying neurons. And so in those develop during a certain cell division, or start to develop at a certain cell division to become those neurons for egg-laying. Now, the male doesn't lay eggs, so it doesn't need those neurons. So during that same cell division and development of those neurons in the male, there's programmed cell death, so those neurons never develop. So these changes then cause differences between the male and the hermaphrodite, so that they have specific functions that they need, and these have all been determined by the evolution of those organisms. So now if we go to a more complex organism, that is fruit fly, Drosophila melanogaster, its process of development is even more elaborate than the nematode. So it starts with a fertilized egg here. There are certain nuclear divisions at first, and then it goes through pupation and eventually metamorphosis into adult male and female flies. This just shows some of the characters of the fly, and then this figure here shows different genes being turned on and turned off at a specific stage in development. So this is that life cycle blown up a little bit. Here's an adult fly. It's colored based on what Hox genes are turned on, and I'll talk about that in a few minutes. But even in the oocyte, there's a differentiation from one end to the other in terms of the maternal products, the proteins, etc., that are distributed throughout that egg cell. Then the sperm fertilizes the egg, becoming the zygote. And then there are 9 nuclear divisions in that cell. So it's a single cell with hundreds of nuclei at that point, called the blastoderm. The nuclei then migrate out to the periphery of that cell, and then new cell membranes are laid down around those nuclei. So now you have cells around the periphery and pretty much a hole in the middle. There are also pole cells set aside at that point, and those become the gametes. It's very important to put aside especially the egg cells, what will become the egg cells early on, so they don't undergo too many replication and division cycles, which would cause more mutation. So those are set aside very early. And then you get into this gastrulation stage, where the gut is formed and other parts of the organism are formed. And initially, what are called gap genes are turned on, and those set up rudimentary segmentation of the animal. And then the parallel genes are next to be turned on, and those set up more segmentation in the animal. Next, the segment polarity genes are expressed, and those more or less bisect the previous segments that are set up. And then there's expression of the homeotic genes at that point, and that pretty much sets the stage for locating legs and wings and eyes, etc., in the adult animal. At that stage, it's a larva, and so it feeds in 3 stages, getting larger and larger. Looks almost like a little caterpillar or slug at that point. And then it pupates. And inside the pupa, certain cells are set aside called imaginal discs that start forming the different tissues for the adult animal. And many of the other cells are just used as energy or resources, that is food for the other cells to grow into those pieces of the mature adult. So then you have emergence of the adult fly from the pupa, and then mating again to repeat the cycle. But as you can imagine, there's a lot of coordination that has to go on between the cells and the expression of the different genes in order to produce a fully functional, mature, reproductively capable adult fly in this case. And incidentally, you have some of the same types of genes in you that led to complete development, functional development of you, each of you. So just to show you a few details of some of the things going on in that organism, if you look just at the egg cell, even in the egg cell, there is a difference between the posterior and anterior end as far as what's being transcribed and what proteins are accumulating in each portion of that egg cell. So here you have transcriptional control, where the nanos gene, or at least the gene product, the RNA, appears to accumulate only on one end of the egg, whereas the RNA for the hunchback gene occurs throughout the egg cell. But then if you look at protein expression, the translation of that RNA, nanos is accumulating near the posterior end and then tails off towards the anterior end. And hunchback translation occurs mainly at the anterior end and almost not at all at the posterior end. So there are gradients set up in the egg cell even before fertilization. So if you look after fertilization, how some of these genes are turned on, both the maternally expressed genes and the gap genes, in 2 insects. One is fruit fly and one is wasp, just to show you that morphologically they are similar, but then they're very different in what they do and how they interact. So everything in blue here is from Drosophila melanogaster pattern, and in red is for the wasp pattern, and then everything in black is the same for both of them. And so you can see there are some similarities in the nose and the hunchback and some of the other ones, but then there are some distinct differences between the Drosophila and the wasp. And then by the time you get to gap gene expression, which happens a little later, then you see more changes, more differences in different genes and proteins that are expressed along those organisms during their development. So you can see, even though they have similar genes and they have analogous genes, that is, the hunchback is the same in both of them, and some of the other genes are the same in both of them, there are subtle differences leading to slight differences in the expression of those genes, and that then makes them have different, slightly different morphology and also slightly different behaviors. But you can see the similarities that join them together evolutionarily by having very similar genes but slightly different versions of each and different expression patterns for each. In Drosophila, this shows a bit of the detail about the maternal gene products, gap genes, parallel genes, etc., and how each one of those gene products affects expression of other gene products. So then you finally get this segment pattern and eventually legs and wings and things like that formed on the animal. So up here, the CAD, bicoid, and NOS or nanos genes, the products, nanos actually inhibits expression of bicoid, and bicoid inhibits the expression of CAD. And so this sets up some of that segmentation or gradient, the gradients within the egg cell. And then these affect some of these other genes that are turned on, which are the gap genes that start forming the segmentation in the animal. These then have effects on the next set of genes, the parallel genes, essentially bisecting the segments and then further bisecting the segments for the segment polarity genes. And then the homeotic genes start taking effect. And the homeotic genes are regulatory genes. So they control the expression of a large number of other genes. So a homeotic gene doesn't make a leg. What it does is it will signal other genes together, and together those genes, as they're expressed, will form the leg. And the homeotic gene won't make a wing, but again, it'll turn on a set of genes that then their products will produce a wing. And they will place them in such a manner that they're in the spot where, functionally they have a use. So a leg coming out of one part of the organism will act like a leg, but a leg coming out of its head won't act like a leg. So homeotic genes and mutations in those can cause very weird types of mutations. And as I indicated before, these homeotic genes are evolutionarily related altogether. So up here you have fruit fly, flower beetle, the arthropods, nematodes, polychaete worms, echinoderms, other types of worms, and tunicates. And then so these are the chordates here, the tunicates, lanceolets, sharks, fish, and mammals down here. And if you line all of those Hox genes up, you can see how they line up, and they line up in all these organisms in an anterior or head to posterior in arms, legs, and tails fashion. So overall, they've been conserved in all these organisms through evolution, but also their order along the chromosome has been more or less conserved, although there are a few inversions here in a nematode and here in a sea urchin. There have been some inversions of some of those genes. And furthermore, they're turned on in the same direction also. So you start on the left-hand side here and move over to the right, turning them on sequentially during development. So also note that all of these ones down here just have a single locus for the Hox genes, but then by the time you get to elephant, shark, and mouse, there are 4 copies because there have been 2 rounds of whole genome duplication. Now there's 4 sets, and then in fish, that is in ray fin fish, there's been another whole genome duplication, so there are 8 versions of each of the Hox loci. Although if you look carefully in zebrafish, one of the D loci has been lost. So there's 2 A's, 2 B's, 2 C's, but only 1 D. And then in pufferfish, it's lost one of the C loci. So there's two A's, two B's, two D's, but only one C. And this is pretty common in evolution, that sometimes you will lose certain genes after a duplication as well. So, and I mentioned this before, where you have the Hox genes, there's an ancestral chordate, you get duplication, and then in jawless fish, there's divergence of those, and then another duplication event to become the cartilaginous fish, as well as mammals, and you get more divergence, and then another duplication event and divergence, which formed the ray-finned fish patterns. This figure shows a little bit more information about how many copies of each of the Hox genes that you can find in different animals, as well as whether there are any homeodomain genes and proteins similar to Hox in other organisms. So if you look back into bacteria, archaea, and protista, protists, there are not any known genes with these homeodomains like Hox genes. This might partially explain why those organisms, those groups of organisms, don't have many multicellular species in them. But then by the time you get to plants, Archaeplastida, they do have homeodomain genes, although they're not exactly like Hox genes. They do have a homeodomain, but it's a different one than found in animals. But it performs some of the same functions, that is, of providing genes that are that control other genes, that control things like petals and leaves where they're put on the plant. There might be homeodomain genes in amoebae, but it's unknown. Probably in some things like slime molds that are multicellular, there probably are some sort of homeodomain type genes. There are homeodomain type genes in fungi, and so that's known there, and many fungi are multicellular. In fact, most are. And then you go up into animals where you have true Hox genes, and this starts even from porifera, which are sponges. Very simple organisms, essentially have an inside and an outside, but they do have Hox genes. So that probably represents the rudimentary gene or gene type that formed all of the other types as well. And then you go into Cnidaria, which are the anemones and hydras and things like that, and there are more, and they have more body parts. And then into Platyhelminth's worms have a few more of those genes and more body plan than, for instance, sponges. Nematodes are a little more complex, and then you get to arthropods where they're more complex and have more versions of those genes, continuing up to echinoderms like starfish, and those have even more of those genes and more complexity in their body plan. And then into the chordates up here, including tunicates, which are simple chordates, but they have almost every type of Hox gene as we have. And then finally up into the vertebrates with birds, mammals, and fish, and they contain even more versions of the Hox genes to determine body plan. These last two columns indicate the number of loci for the Hox genes, and then the total number of Hox genes themselves. So you can see as you start from porifera, where it's quite simple, moving up into arthropods, where there's now been a duplication, so there are at least two versions and more genes. Finally up into the mammals, where there are four loci, due to whole genome duplication again. There would be 8 copies in fish here, Danio, which is zebrafish, but one locus has been lost, so there are only 7 versions, but 49 Hox genes, so a lot of Hox genes, especially compared to porifera. But then that explains the complex body type also. And again, just looking at this phylogram again that has superimposed on it the type of appendage plus the number of Hox genes, and you can see how you change from a large number of bones in the fin and then the fin itself, fewer bones here and more of a paddle foot, and then finally a foot with fewer bones and then fewer bones in mammals, and the fewest of all in birds. This correlates well with Hox D13, and Hox D13, when it's at low levels, you get more bones formed in the hand or fin. As Hox D13 has increased in expression, there has been a change from a ray fin, or a fin with a lot of bones in that fin, to a lobe-finned fish with fewer bones, to something that looks like a reptile, a pre-reptile essentially, and then fewer yet in mammals and birds as the Hox D13 levels have increased. Experiments with mutant mice in the Hox 13 gene, as well as a different gene called GLY3. If you have a mouse with wild-type Hox 13 and wild-type amounts of GLY3, then a normal foot is formed with 5 fingers on it, or 5 toes. As the levels of Hox 13 are reduced to 0, actually, then a club forms rather than a foot. The number of, or the amount of GLY3 determines the number of fingers or toes. So that if you have a wild type, then 5 toes are formed. If you have a totally mutant level of the GLY3, then more of the toes are formed, more bones and more toes. And so if you have the double mutant, then you have a larger club with a lot of bones within it, as if there are going to be more toes. So for these regulatory genes that are controlling a large number of other genes, then in a very short amount of time, in a generation or two, you can get very large changes in some of the morphological characters on those organisms. So depending on where and when those genes are turned on also affects the morphology of the organism. So here we have a differential expression of Hox 4, 6, and 9 in neuronal tissue, and Hox 6, 9, 10, 11, 12, and 13 in mesoderm. So up here will form the neurons coming out of the spinal column, and here will form either the fin buds or the limb buds. And you can see in fishes, those genes are turned on higher in the spinal column. If that happened in you, then you would have your arms coming out of your neck somewhere. In fish, then, their fins are really a little further forward on their body than they are in mammals. Also note that the neuronal tissues and the fin bud itself and the mesoderm are coordinated. So you have the muscles in the fin bud and the fin being coordinated with where the nerves are coming out to innervate those appendages. And in the tetrapods, like mammals and similar things, right, you have the arms and shoulders coming out further down the spinal column than in fish. Both the nerves and the limb buds are coordinated so that they come out in the same place, so that you have a limb, an arm, let's say, that comes out in the same place where the nerves are coming out to innervate that nerve, so that innervate that limb, so that the arms and fingers and things work properly. So some of these major differences between fish and tetrapods and insects and other things are due to differences in the expression of those Hox genes and other types of genes when they're turned on during development, where they're turned on during development. So you can get large differences in these morphologies of these organisms over relatively short time spans evolutionarily. And remember, these are genes that have been conserved and passed on through different lines and different species and different groups of organisms, but just have different patterns of expression leading to the different morphologies of those organisms. So just to show you a few examples, these are the patterns in the neuronal tissue layers, the things that will form the neurons. Now, in your bodies as well, you have ectoderm, endoderm, etc., and several layers of each actually. So you have different layers that will express these genes differentially. So, for example, in tetrapods, these Hox genes are turned on a little lower in the neural cord for both the hands and arms, or front limbs and hind limbs. And then they're turned on a little further forward in fish. And then in tunicates, they're turned on in a different pattern, and there are fewer of them. And then in the hemichordates, they're turned on, but there's a little bit of, instead of being turned on or off strictly, there's a little bit of leak through to different tissues, so that the form of these animals is a little more gradual. And then in the arthropods, this is the pattern. Arthropods have a longer posterior end than do the tetrapods, and that's true. As you can see, these longer production of tissue and neurons in this case the abdomen. And here are the patterns for somatic tissue, that is the tissues that form the muscles and things like that, the non-neuronal layers. They're turned on in the same places as the neuronal layers in tetrapods and fishes, and then a little bit difference in hemichordates, and then this is in annelid worms. And so in worms you can see there are longer patterns, and you can sort of guess from this that these are extended organisms, longer organisms. And then again in somatic tissues in arthropods, you see this extended posterior end, which is part of the abdomen. And then these are Cnidaria. These are fairly simple animals. These are anemones, which have just a few Hox genes, and primarily they have a lower portion to their body, an upper portion of their body, and a mouth, and that's pretty much the entire animal. Just to show you one of your long-lost ancestors, this is a tunicate. It's also a chordate. You're a chordate. You're in the phylum Chordata. But this is one of the very earliest chordates, and essentially it has this part here, which is the body. And so you can see the Hox genes that are turned on there in the epidermis, in the muscle, in the neurons, and the notochord. And then this is the notochord with the tail. You also had a notochord when you were very early in development, but it was pushed aside by the spinal column, and so that's how you became a vertebrate, having that difference. But you can see the different parts: the heart, the stomach, the intestines, ovary, testicle, and the patterning of those Hox genes, although a simpler set than what we have. And so this is a really simple phylogram showing the Cnidaria. These are the anemones with a top part of the body, a bottom part of the body, sort of a holdfast, and a stomach interior, and a mouth and some tentacles with very few Hox genes. And then here's a nematode, again with fewer Hox genes, and how they're turned on to form that body plan, and then eventually extension. The arthropods are more complex, and leads to also segmentation of the body more so than other organisms below that. And then cephalochordata has a simple spinal column type pattern, the first of the chordates, and just a little more advanced than the tunicate that I showed you in the last slide. And then finally, the chordata, which is us. And you can see that there are several layers in humans and other vertebrates, so that each cell layer and each histogenic layer essentially forms a slightly different type of patterning. And from this, you can also see how each of the Hox genes is turned on or turned off, or a combination, going from the head, where these are towards the anterior portion of the organism, and the posterior end of the organism, and the same going from HOX1 down to HOX13 or HOX14 down at the end. And this is how they're expressed in a hind limb or a forelimb. So starting in the bud of that hind limb or forelimb, HOX9 turns on, and then a little later, HOX10 and 11 turn on, forming a little more variation in those tissues and cells. And then HOX12 and 13 finally start turning on, so that by the time you have a forelimb or a hind limb, the expression is this, where HOX9 is responsible pretty much for forming the shoulder and shoulder blade, HOX9 plus 10 for the upper arm, 9, 10, and 11 for the forelimb, 9, 10, 11, 12 for the wrist, and 9, 10, 11, 12, and 13 for the hand and the foot and the fingers and toes. And it forms those in order, so you'll have a thumb on one end going all the way to the little finger on the other side. And remember, these genes are responsible for turning on and off other genes to make those different tissues. They don't do them on their own. They don't form them on their own. And the system is a little more complex than even that, because there are enhancer elements, EE, and global control regions down on the other end. And so these ones are actually turning on the Hox genes, which then are turning on other functions as well. And so the enhancer element will turn on these early Hox genes 1 through 4, and then actually 1 through about 10 in order. And it has less and less influence the further down this piece of DNA that you go. And then on the other side, this global control region controls these Hox genes in later expression, so that you get them turning on from 1 all the way to 13 and 14 if there is a 14. And then this is how each one of those is responsible for those different regions of turning on in both the limb bud and then eventually the limbs. So the sequence of evolutionary events then is thought to go something like this. You have originally one Hox gene or one homeodomain gene. You have duplications of those, and then they'll start acting in different ways in those organisms, forming different tissues. You have more of those, and eventually have local control elements controlling each one of those that can give you finer distinctions in how to form those tissues. Then eventually there are controlling elements either upstream or downstream, that begin to control these elements in a different way, in a more coordinated way, as the local elements, local control elements, lose their influence over that section. And then eventually you have more and more control. Some of these genes disappear, and you can get different versions of them to form different types of tissues, different types of organs, placement of the organs, and have some very complex body plans because of this. And so these are some of the things that really had an influence on making body plans for complex multicellular organisms. Okay, so that's the end of this introduction to multicellularity and cell communication. In general, then, multicellularity is partly due to the evolution of these controlling agents or controlling elements that then began controlling body plan, that is, controlling when and where genes were turned on, where they were turned off, and what sort of appendages, neurons, leaves, and I'll talk about plants in another video, and other organs were formed. And it led to a lot of different body plans that we see today, a very diverse set. But this can only happen with these gene duplications, followed by diversification, and then high degrees of control in expression of those genes. Okay, so that's it for now. Thanks, and I will talk to you later.